Okay, we're nearly at the end now. This is going to probably be the last tricky topic that we're going to be covering for this course for the Year 2 Genetics. We have another one or two after this, but they're fairly easy and fairly fun, so don't worry after we've done this. Last time we talked about how the agrobacterium are able to use their TI plasmids to take their tDNA and insert it into a plant cell genome via use of the Vir operon. Go back and watch that video if you haven't watched it already, because I'm going to assume that you have, and you know how a plant cell actually, how the Vir operon functions to insert the tDNA into the host plant cell. We're going to expand on that knowledge today. So first things first, the Bin19 vector, which just is short for the binary 19 vector because we're using a binary system with the R and the agrobacterium that we're producing through this process is going to have two plasmids, so it's called a binary system. So the Bin19 vector, part of the binary system, the Vir operon genes in the binary system come from the agrobacterium TI plasmid, and they are contained within what we call a PAL4404 plasmid, and the tDNA genes come from a second plasmid from our E. coli that we just call the transgenic Bin19 plasmid. So the Bin19 plasmid is a composite of these. It has, the one we're talking about, it has a left and right border and the tDNA. We've got a picture of it on the right, so you can see the left border is the green box, the right border is the right box. The tDNA is in between that. We have the LacZ gene in between it, which we're going to explain in a moment. And we also have the CAN-R gene, which is going to be important later because it gives resistance to an antibiotic called kanamycin, which we need for when we are doing, from when we're separating infected plants from uninfected plants. So, just look at the image on the right. The transgenic plasmid that we're talking about has the left border, the right border, the tDNA, a LacZ gene, and we're going to insert a plasmid inside of that LacZ gene. So, sorry, we're not inserting a plasmid, we're inserting a gene of interest within the LacZ gene. So say we want to produce a plant that can produce more anthocyanin, we want to add an anthocyanin gene into it. We put the anthocyanin gene within the LacZ, in between the borders, and then we have a separate plasmid because we have two, because it's a binary system, in the PAL4404, and that PAL4404 plasmid contains the Vir genes from the Vir operon. So it's a bit of a mouthful. So we have a polylinker within the tDNA of the Bin19 plasmid, which is a short sequence of DNA containing the recognition sites for restriction enzymes. The polylinker has these restriction enzyme recognition sites inserted in a sequence, so all we have to do is open up a space between two of these sites and insert our desired gene product. So we can see the polylinker labeled on this Bin19 plasmid here as a bunch of kind of upside down L's on top of the LacZ gene. These are all restriction enzyme recognition sites. We've basically taken two of these recognition sites, we've used a restriction enzyme to cleave them, to open up the DNA, give it sticky ends, so we can insert our LacZ gene. So the LacZ gene has been inserted within 2 of these restriction enzyme recognition sites within the polylinker. So why have we put the LacZ gene inside of the polylinker? We use a selection system to separate E. coli that take up our transgenic Bin19 plasmids from those that have not taken up our transgenic Bin19 plasmids. So basically, the LacZ gene codes for an enzyme called beta-galactosidase. It's inserted within the polylinker, like we just discussed. When the plasmids are spliced and we insert our desired gene, so for example our anthocyanin gene, within the LacZ gene, it makes the LacZ gene dysfunctional, so it can no longer produce this enzyme. And if the plasmid can't produce this enzyme, it can't break down its substrate. So when we have all of our agrobacterium, we're trying to inoculate them with our plasmids. So when we make our plasmids, not all of the plasmids are going to be transgenic. Not all of them are going to be cut in the middle of the LacZ gene, and not all of them are going to have our trans gene. So not all of them are going to have our anthocyanin gene in our example. If they don't have our inserted gene within the LacZ gene, they're able to break down the substrate of the XGAL called beta-galactoside, because they are able to produce the beta-galactosidase enzymes. But if we have our inserted gene within the LacZ gene, they can't produce that enzyme, so they're not able to break down the beta-galactoside in the XGAL inside the XGAL substrate. So if we look to the petri dish on the right, we can see that some of the bacterial colonies of the E. coli are blue and some of them are white. The blue colonies have basically, sorry, one sec. They're being cultured on a substrate called XGAL. The XGAL contains beta-galactoside sugars. These beta-galactoside sugars are broken down by the beta-galactosidase enzyme produced by transcription of the LacZ gene. So if they are blue, that means they have broken down the substrate, which means they have active LacZ genes because they're producing the enzyme. If they have active LacZ genes, that means they haven't taken up the gene that we've inserted, because we're trying to insert the gene within the LacZ gene. So if they're blue, they've taken up one of the plasmids, the Bin19 plasmids, but they haven't taken up a Bin19 plasmid that has had our desired gene inserted within it. So they're no good to us. We only want the white E. coli colonies, because the white colonies are not able to break down the beta-galactoside sugars within the XGAL substrate, because they have a gene within their LacZ gene that we've put there, so they're not able to produce the enzyme, they're not able to break down the substrate, they're not able to change colour. I had hoped we'd get further than that, but this has taken a while to talk about. So far, we've gone over what the Bin19 plasmid is and how we insert a transgene within the Bin19 plasmid, so the gene that we want to express in the plant. And we've talked about how we separate E. coli that have taken up are transgenic Bin19 plasmid from those that haven't.